Hi everyone, this is Kumar. Faculty of Pharmaceutics Department belongs to SMS College of Pharmacy and Health Sciences. Today our students are going to speak about the importance of the sterility testing that is purely relevant to the human centered. So you know the meaning of the human centered. So what do you mean by human centered means where we are going to apply the sterility testing of pharmaceuticals relevant to the pool or the profession where they involve. For example, patient, pharmacist, industry microbiology area, almost all area personals they are involved in. So in order to appropriate sterility testing of pharmaceuticals we need. If you colloquially take sterility testing means it varies from one field to the another field. Sterility testing of pharmaceuticals means it's only applicable to the pharmaceuticals area. So sterility testing applies so many wide areas like laboratory, hospitals and another water treatment plant, so on. So here we have to approach in the human center. The common concept is nothing but to identify the pathogen and remove, but the human centered and the areas will be vary from one area to another area. Here our students they are going to talk about the same heading under the design thinking parameter. They will come and introduce themselves and they will take one topic and they will cover up.
I am Jeevan. I am talking about empathize regarding sterility testing. Empathize. Understand the needs, problem, and the experience of the users. I am Jeevan. I am talking about empathize of the sterility. Empathize. Understand the needs, problems, experience of users. Observe how sterility testing performed in laboratories. Identify difficulties such as contamination risk, complex procedure, or time-composing steps. Collect feedbacks through interviews, observation, and discuss. Example: understanding the challenges faced by microbiologists while handling sterile samples.
I am Praveen. My part of the sterility testing is nothing but define, clearly identify and define the main problems based on the information collected. Next, frame a problem statement for sterility testings. For example, laboratory technician needs a faster and contamination-free methods for sterility testing to ensure safety pharmaceuticals products.
Hi, I am Arun Pandyan. My part of sterility testing is nothing about idea. Generate many possible ideas and solutions for the defined problem. Brainstorm innovative ways to improve sterility testing. For example, automated sterility testing system, improved sterile workshops, faster microbial detection methods, and simplified testing protocols.
Good morning, I am Santoshi from B.Com Thurston. Now I am talking about the prototype of the sterility testing. Prototype. Simple models or trial versions of the selected idea. Prototypes can be new sterility testing devices, modified lab procedures, software systems for monitoring contamination. Purpose: to check whether the idea works before full implementation.
I am Kamachana. I am studying in SMS College of Pharmacy and Health Science. I talk about test. Evaluate the prototype in real laboratory condition. Check accuracy of sterility result, time required for testing, easy of use for technician. Collect feedback and make improvements.
So we are going to sum up the design thinking of the sterility testing. So as they discussed, so it should be vary from one department to another department and vary from according to the traditional and modern. If you take the modern, now the modern is nothing but an automated system. So automated is nothing but the composition of the appropriate sensor towards the pathogen, non-pathogen. So we have to identify in which lab as a more pathogenic microbes. According to the pathogenic, we should develop the automated system in order to quickly evacuate or eradicate the microbes. The first thing is identify the suitable biosensor towards the particular strain. You know the strain. So in one genus and one species there are so many strains. and almost you know the meaning of the mutation, how they will get mutated, how to identify the mutation, all are based on the sensor based and automated based we have to set up. So automation is nothing but a combination of the electrical as well as the communication and as well as the microbial impregnated, it's nothing but a embedded technology. So you know the meaning of the embedded technology, what are the components of the embedded, it's nothing but a electrical sensor, component of the microbes as well as the bio. So combination of these three you can identify with the help of the AI and simulation. So what type of microbes, whether they will get the mutated or not, we can easily identify. So this is somewhat the typical area, especially exclusive for the high pathogenic area. Now we are going to discuss about the same concept towards the less virulent areas, nothing but here small laboratories and small scale hospitals. Likewise, there the population pool will be very less. So what type of populations usually come? For example, if you take a pediatric division, there is only pediatric they are going to concentrate, more than that they are not going to evaluate anything. So there we can expect a low microbial pool only. So for such type of area, there is no need the advanced identification, strain improvement like automation technique. As usual, conventional, it is so safe for it. It's a convenient as well as the time consuming as well as the cost effective method. So here we have to identify first of all whether the particular concern or organization or the sterility area need a high level automated or the as usual the conventional. Based on the budget area we have to design the sterility testing of the parameters. So likewise the students what they delivered, likewise what I am going to cover regarding all those areas. Based on overall our ideas, we hope that you will get some about the sterility testing of the pharmaceuticals where we can apply, where it is essentially needed. Thank you.